In this video, we will talk about gut microbiota and in particular about dysbiosis and multiple sclerosis. When the intestinal microbiota of healthy individuals has been compared with the ones from patients affected by multiple sclerosis, some significant differences have been observed, which indicate that dysbiosis often exists in people affected by this disease. In this video, we will review some of the first studies which have been published about the intestinal microbiota in multiple sclerosis. The first that we will comment on examined the intestinal microbiota of 7 persons with MS. Of these, 5 received a treatment and 2 not. Further, they studied 8 healthy controls who did not live with the patients, and all were white persons. And in the intestinal microbiota of the persons affected with MS, some bacteria were found to be increased, among them Akkermansia. In the second study, 20 persons with MS were analyzed, and 13 of these were on treatment, 7 not. It also included 50 healthy controls not living in the same household, and all were of Asian descent. And again, certain bacterial strains were identified that were more abundant in persons with MS. Some of these bacteria matched with those in the previous study. However, in this study, Akkermansia was not among them. In a third study, 31 patients with MS were examined. 20 of these followed a treatment and 11 not. The study also included 36 healthy controls who did not live in the same household. And again, several bacteria were identified that were more abundant in patients with MS, although the types of bacteria that they identified were different from those in the 2 other studies. In a fourth study, 60 persons with MS, 32 on treatment, 28 not, and 43 healthy controls not from the same household were investigated. And here again, Akkermansia was the most abundant or most different bacteria in patients with MS. Other later studies also documented higher abundance of Akkermansia in persons affected by MS. In the first of these, 18 patients with MS, 9 with and 9 without treatment, as well as 17 healthy controls, not from the same household, were studied. Also, both persons with white and black ethnicity were included. The second study looked at 71 untreated persons with MS and 71 healthy controls from the same household, and all were of white ethnicity. And in the third, 34 persons with MS, 19 of whom with treatment, 15 without, as well as 34 healthy controls, all homozygotic twins, which means genetically identical to the patients with MS, were investigated. From all these studies, Akkermansia emerged as one of the bacteria that was most frequently found to be increased in the gut microbiota of persons with MS. As we previously commented, these are only a few of the first studies that had been published. There are more and more studies not only about gut microbiota in general, but also about gut microbiota in MS. And the majority of the studies that have been published until now support the relevance of Akkermansia in MS. The intestinal microbiota has important functions in the digestion of food, the defense, the communication between intestine and brain, and the formation of the immune system. The gut microbiota digests the remains, leftovers of food that reach the colon, which could not be digested in the small intestine. During the microbial digestion in the colon, which is called fermentation, molecules such as vitamins and short-chain fatty acids are produced, which are beneficial for our organism and pass into the blood. The gut microbiota also participates in the defense against pathogens, that is, infectious agents, by covering the mucosal surface and preventing that it is colonized by these pathogens. The gut microbiota can also influence the information that reaches the brain and therefore our subconscious. And finally, the gut microbiota also plays a role in the formation of regulatory immune cells, which are required for the development of an efficient and balanced immune system. If we have dysbiosis, which means an altered gut microbiota that is not able to fulfill these functions in an optimal way, we will expect poor digestion at the level of the large intestine, the colon, and hence lower production of vitamins and short-chain fatty acids. an insufficient protection of the intestinal mucosa and therefore higher probability of disruption of the intestinal barrier as well as intestinal infections. Alterations of the gut-brain axis and, as a consequence, changes of our subconscious and the perception of pain, emotions, stress, and fatigue. And finally, an immune system with deficits of regulatory cells and mechanisms, and, as a result, higher likelihood for the induction of allergies and autoimmunity. If we have dysbiosis and multiple sclerosis, people affected by this disease should present all or at least some of these alterations. And in fact, it has been observed that in people with MS, the concentration of short-chain fatty acids in serum is lower than the concentration in healthy controls, suggesting a lower fermentation at the large intestine level. In the serum of people affected with MS, it is also common to find markers of alterations in the intestinal barrier and markers of the presence of microorganisms. Moreover, these markers correlate with disease progression and disability. People with MS also often suffer from fatigue, depression, and pain, which may be related to an alteration of the brain-gut communication. Finally, MS is an autoimmune disease in which regulatory immune cells are deficient. Altogether, patients with MS may show all these alterations that are associated with intestinal dysbiosis.